And here the agenda of this webinar. So this afternoon, I will start introducing to you with EATRIS, because I'm not sure that everyone is familiar with this organization, this research infrastructure. Then I will give the floor to Sven Parkel, General Manager at Turtu Biotechnology Park and partner in the BIC Bridge project, and he's going to introduce you to the BIC Guide. That is a very nice tool developed within the BIC Bridge project context and focus on biomarker commercialization. And finally, Anton Ussi, the EATRIS Operation and Finance Director, is going to present you a newborn project, is a proposal still, the European Biomarker Accelerator Fund. Sorry that people are joining. Okay, so today, I will start introducing you to EATRIS. EATRIS, as mentioned, is the European Infrastructure for Translational Medicine. We are a research infrastructure of over 100 research institutes from 14 European countries that via a central hub deliver research services to researchers, companies, founders, and charities to help translating the scientific discovery into benefit for patients. Our research institutes are organized in 5 scientific platforms, and we can provide high-end facilities and expertise in different fields. So as you know, the translational pipeline represents a very strong operational challenge, and this process is defined by a complex interaction of different players. We have researchers, but also clinicians, patients, policymakers, industry, regulatory people, and the success of these people work can only occur through a structural and cross-sectional collaboration. And in order to accelerate the translation of research discoveries into patient benefit, EATRIS supports these stakeholders using the capacities and expertise present in the institutions that are part of the infrastructure. At the heart of our community, that is multidisciplinary and very committed, there is the patient. So in fact, our mission is to improve the development of high potential medicine and diagnostics that meet patients' needs and positively impact their health outcomes. As mentioned, our research institutes are organized in 5 scientific platforms that can provide really a lot of expertise, but high-end facilities as well in different fields that you can see listed in this slide. So we have capacities within the advanced therapy medicinal products, biomarkers, imaging and tracing, small molecule and vaccines. So through the platform, we provide academics and industry with cutting-edge academic expertise and technology services, selecting scientific priorities for developing new tools, developing educational and training initiative for educating the new generation of researchers. And finally, our final aim is to build an effective translational medicine ecosystem. So we are also involved in different European projects, and among them, the BIC Bridge project. We joined almost 1 year ago as a full partner, and there our role is disseminating the tool that has been developed previously in by the consortium partner. Here I would like to thank the member of EATRIS, and if you want more information about us, please stay in contact. Now I would like to give the floor to Sven.
Thank you, Emanuela.
That is gonna actually go a bit more in detail on what is the BIC Bridge for you and the added value for you.
Excellent.
Thank you, Sven.
Thank you. Thank you so much. I hope you can also see my slides.
We can see it maybe in presentation.
Now it's better,
Perfect.
I hope. Excellent.
Yeah. Thank you.
So many thanks for the invitation. It's a great pleasure to be here and to have a discussion with EATRIS on the biomarker commercialization and development. So you may notice that I'm not Valerie. Valerie unfortunately could not be here today. I'm Sven Parkel, as Emanuela already said. I'm the general manager of the Tartu Biotechnology Park, and we've been involved with the project from the very start, from the very beginning. Just 2 words about my background, so you would know where I'm coming from. My background is in chemistry, but I've also done 4 years in the University Tech Transfer Office, leading different biomarker projects, among others, and now for 3 or 4 years now been running Tartu Biotechnology Park, and for a couple of years also the international network Scanbalt. So that's my current position. But go back to the biomarkers. So we have quite a few sort of general background information slides. I'm not sure how relevant those are for you, but I'm just gonna make a quick overview anyway, because I don't know how familiar you are in general with the biomarker and with the biomarker development process. So bear with me if this is too common knowledge for you, but hopefully not. So you may know that about 60 to 70% all critical clinical decisions are made by test results, so some kind of lab test results. And as you may know, the biomarkers are behind those kind of lab test results. And the biomarkers market is growing heavily, as there are new biomarkers coming out quite regularly, but not as often as we would like to see that. And but yes, more and more of the clinical decisions are moving into being reliant on some kind of lab test, which is again relying on some kind of biomarker. And when we talk about biomarkers, then it's important to keep in mind that there are 3 sort of main aspects regarding biomarkers. So one of them is specimen. There has to be some kind of sample, either it's a blood sample or a skin sample or something else, where that can be drawn from the organism that is under study. And another important characteristic of a biomarker is quantification. So the biomarker has to be quantifiable, and of course there has to be the context. So the amount of the biomarker should be dependent of some kind of physiological process, and especially it becomes relevant in the context of disease if the amount of the biomarker is somehow related to the disease progression, or we will take a look in the next slide, I think, in other aspects of a disease. But here are some definitions of biomarkers that you may have come across, but in general they take into account all these three aspects of biomarkers. And when we talk about the biomarker invention or biomarker discovery, and then this is sort of the aim of the BIC Guide, is to support the overall and better discovery of the biomarkers, is that the biomarkers can be used for diagnostics. It means that to test whether there is disease or some kind of physiological process that could be relevant to a disease. It's for drug discovery and development, of course, for personalized medicine, for disease risk assessment, so let's say some kind of genetic markers here, and of course for other applications, let's say DNA fingerprinting and other opportunities as well. Sorry, I have lost. Yeah, okay, now it changes. So the aim of the project, which used to be a big project in the first phase, and which is now the BIC Bridge project, is to support the bringing of the invention from the discovery to a product on the market. And if you have worked with or been involved with biomarker projects, you know that it's rather extensive, and there are several aspects to be considered. But what is mostly important here to realize is that while research is extremely important, the benefits for the patient start only if there's a commercial route and the biomarker reaches the market in a form of a lab test that can be reliably used to estimate some kind of aspects of the disease. And most often the goals or the views why these kind of novel tests are developed is, of course, to provide better or earlier diagnostics, to predict the risk for development of certain conditions, to prognose the development of a disease, or monitor the effect of a treatment. Let's say if you have developed a very expensive cancer treatment and the reimburser wants to know whether this cancer treatment actually works, you have to figure out a way to show that the treatment actually reduces the disease condition. So why a BIC Guide? So I already hinted a little bit within the previous slide framework, but what we quite often see is that most researchers don't have a good hands-on experience with commercialization. And while researchers are extremely good in knowing the technical requirements for biomarker development, quite often the regulatory side and the industrial aspects are not generally the focus. And what is really important in the biomarker projects is that already quite in the very early stages, the researchers would be aware of the regulatory side and also of the industrial needs. That, for example, the biomarker that they are developing should be testable in a high throughput format. Let's say if you have to run each test manually for a long time, then this is rather time consuming and perhaps could be let's say inhibiting the overall use of this biomarker in a larger frame. And from the company perspective, they mostly rely on the market and clinical needs as well as the cost-benefit analysis. And here we reach the question. So let's say the researcher has developed a biomarker, but what does the industry need? So when are the companies willing to collaborate? How can I take this biomarker to the market? What do they need? Is it the proof of principle? What kind of validation is needed? What should be included for a proof of concept? So those are the development gaps that we've been trying to address with the BIC Guide. And so the aim is to create an overview of the process behind the journey from discovery to commercialization. So it's sort of the support material. It goes or is aimed mainly to the users. It's a team comprising scientists and TTO at least, but why not also other stakeholders in the whole process? And the aim of the guide is to support the maturing process of the commercialization. And the technology transfer from research institution context. So of course the fields are broad and there's a diversity of nature of medical devices. So it could be quite difficult for a TTO to keep all that in mind, especially if they're not working with the biomarkers on a daily basis. So technically, the BIC Guide is a supporting tool for evaluation, development, validation of biomarkers, inventions into commercial products. So if you are sure that your biomarker does not have a commercial value, then perhaps the guide is not for you, because the aim of the guide is to mainly support the commercial route, and it's been set up in this way. Of course, you might find use from the BIC Guide either way, but it's just important to notice that it's mainly for the commercial route. It is most important to stress here that the guide is not here to tell you exactly what to do, because there are so many aspects, and so there are so many different biomarkers. The situations are different, the people are different. There is no one fixed recipe, but rather the BIC Guide is a decision support tool. So it will help you make decisions and check that you have done those things that you should be doing. But yeah. And then it sort of gives you a process, gives you a workflow how to develop your biomarker in a relevant fashion that it would talk to the TTO and also to the commercializing help. So in a way, if you've been involved in tech transfer, you know there's quite a bit of talk about the valley between scientists and industry. So in a way, it helps to improve this communication between the researchers and the industry side. It supports the dialogue and collaboration between the TTO and scientists, of course, because it sort of provides clear paths, clear milestones, clear roadmap how to define and how to develop your biomarker project. It gives better visualization of the cross-disciplinary approach that's very often needed to reach a successful commercialization. There are many different stakeholders and aspects involved, and then we started to look at the perspective from different users, but then decided that it's better to focus on the journey itself instead, and not try to give guidelines for each specific stakeholder group that's involved. So rather we will sort of provide a decision support tool for everyone. And now we reach to the BIC Guide. So as you may notice, I will show you some of the screenshots as well. As you may know, or as you will see, there are 7 phases. So those are TRL based, and we have included 3 tracks. And a list of tasks under those tracks I will show you in a minute. So there are support documents for specific tasks, and then checklist, self-evaluation tool at the end of each phase, and links to useful materials. So you will find the BIC Guide at the address biomarker.nu, and there are additional materials as well that go with the guide. And here's the screenshot of a BIC Guide. So as I mentioned, you have here 7 different tracks. I don't know if you can see my mouse, but mouse arrow, but I'm pointing at the. Oh, excellent! I'm pointing at those 7 phases, and then there are different tracks. So there's technical and clinical evaluation, there's commercial evaluation, and of course the regulatory part. And as you go along under each of these phases, so okay, here's this is in a bigger picture, but now we have moved on from tracks to description and achievements, and here we have described what could be the activities in those phases, TRL-based phases, and what kind of achievements you should be looking at in a given phase. So then the documents, there are some supporting documents that you can either use directly or take as an inspiration for your own activities that support you in a given phase. And then there's a self-evaluation part. So there are some questions you have to think whether you are able to answer them, whether you have thought about your project from this angle as the question asks. And then if this looks good, you should be able to move on to the next phase. So we have tested the BIC Guide already with a number of scientists, and the feedback has so far been very good. So it seems that the scientists are enjoying the help that the BIC Guide provides them. And I'd like just to point out a couple of resources that you can also find at the biomarker.nu address. So this is the best practices handbook, where we have gathered a number of use cases, number of suggestions how you should approach your biomarker project. We have review tool, which provides in a way like a checklist or additional tool to work with your biomarker project that makes it helps you to understand whether you have whether you're progressing in the right direction, and it helps you with a checklist to go forward. And then we have gathered a number of guidelines for regulatory process. A sort of small addition here is that due to the very late, or not late, but recent developments in the biomarker regulatory field in Europe with the new IVDR rules that were announced maybe a week ago, I think, in general. Then some of the regulatory process have not been included in the guide yet, but we're working on that as well, on the guidelines. And here is the summary of this. This is our webpage. Please follow us in LinkedIn. And yes, as the name says in the bottom, we used to be a big biomarker commercialization project. Now it's called BIC Bridge. So that's all from my side. I'm open for questions, for discussions, and thank you all for listening.
Thank you, Sven, for this clear presentation. Are there any questions from the audience? Yes, Uros?
Yes, hello. Thank you for a very clear, nice presentation, introduction to this. A simple question, can be biomarkers patented or only method of accessing them? So, and the second question would be, are there any successful stories that would guide us in this process that you are describing with this tool? Thank you.
So if I did understand correctly, the question, the first question was if biomarkers can be patented. Definitely they can be patented. Of course, it depends on the context in the sense whether they have already been patented or published. So there are different, of course, aspects that have to be kept in mind when you're thinking about biomarker patenting. It's a let's say rather complex process. But I would suggest that as a first step, I would always suggest to take contact with the tech transfer office to see if they can provide you with some general guidelines. So in general, the patenting process is rather common, whether you're working with biomarker or some other type of invention. There are still common best practices how to take a look, and usually the tech transfer offices are competent and can provide you at least the first information about whether it seems, or they can actually take a look themselves whether a biomarker can be patented or not. Now about the second question, are there any success stories on biomarker development? So I'm guessing you're asking that would take from the early stage in the lab until the commercial route, or until actually to be used as a lab test. So there are some. I have heard in the history a couple of them, but I don't know any in the PDF format that I could I could forward you. But usually those can be heard, let's say, in a company presentation if there's a scientist who actually speaks about their story. So I've been lucky enough to hear a couple of this kind of presentation where a scientist has started with a company and is actually presenting the story, how they developed the biomarker into a successful lab test. But these are rare, I agree. So if you're pointing out the sort of lack of the good success stories to learn from, then this is one of the reasons why there is a BIC guide. So we see the same thing, that this kind of success stories are not openly discussed for some reason. Maybe there are agreements behind this that prohibit the scientists to come forward and talk the whole story. Maybe there are some investments that have to be protected, and that's why this cannot be discussed openly. We're not fully sure, but for some reason, I fully agree there are not enough good success stories that would help scientists to go forward with their own invention, and that's sort of why we have developed the BIC guide and hope that this provides value for scientists.
Thank you, Sven. We have an additional question in the chat from Louise at Radboud UMC. Does the guide also provide advice on combination biomarkers?
Combination biomarkers, I would first instance say yes, but then again, maybe if you could ask a more specific question, then I would understand if you mean any specific aspects of this approach. If you could just extend a little bit of the question, because I don't think we were excluding, but maybe there's an aspect I'm not currently recognizing.
Louise, would you like to add something to your question?
Hi, yes, I'm Louise, thank you. Yes, I was wondering about the trajectory for the development of combination biomarkers. Is that the same as for one individual biomarkers, or are there big difference in that? And the same for the regulatory trajectory? Do you need to do twice the work, or is it easier for a combination of biomarkers?
So I haven't worked with combination of biomarkers myself, so I can only give you a feedback from what I have heard and what I think would make sense. Starting from the back, of course, I haven't checked the new IVDR regulation on the combination of biomarkers, so I don't know how much is actually changed there. I have to say that This is something I'm not aware of. When we talk about the general development of combinational biomarkers, then it really depends how much is known of those different biomarkers. So I'm guessing that if either one of them is rather well already characterized, then the workload of the combination is not that big when we compare to just one biomarker. But if we would take that neither biomarker or more biomarkers, they're not known and they're not characterized, and then probably you have to characterize each one separately and then also the combination, I would assume. But I'm guessing there could be some shortcuts. I'm not sure exactly where they would be, but combination is perhaps more difficult unless one of them is fairly well characterized. Then it could be, then there could be easier routes, and then you could just work with mainly with the unknown biomarker and then their combination, and this could reduce the overall workload, I'm assuming. Yeah.
Okay. Yeah, thank you. So we see more combination biomarkers coming up at this time, and I think that will only become more in the future, because of course if you have more biomarkers, then you can probably make better predictions.
Yeah, that's true, and I think the combination can be quite a good recipe in many cases, because finding just one, I mean, what we see at least in cancer is that there is no, I mean, what people who work with cancer say is that there are no cancers that are exactly the same. So all cancers are a little bit different, and they really depend on the people. So it's quite difficult. It's the same with the medicines. So not all medicines work equally for all people, and it's a similar approach with biomarkers I mean, sometimes the disease is very well quantifiable with one biomarker, but sometimes you have sort of those borderline cases or these areas where one biomarker just doesn't give the full picture, and the disease is always or very often more complex than just one biomarker can reflect. So that's why these combinations give clearly a better picture about the disease and the progression and the opportunities that could come with that disease. So yeah, the combinations could be a key going forward. Yeah.
Okay, thank you.
Thank you. So for time restriction, I will go now on. So I will give the word to Anton. But if you have any additional question, don't hesitate to address them or through the chat or reach out us by mail. Thank you, Anton. The floor is yours.
Thanks, Emanuela. So I'm going to talk about an initiative that is in the phase of starting up, and this is actually a call out to you as either researchers or representatives